Does that look good?
That looks great. Thank you. So it's my pleasure to introduce Dr. Taylor Heckman. She's a postdoctoral scholar in the Aquatic Animal Health Lab at the University of California, Davis, where she recently completed her PhD in microbiology. She's originally from Hawaii, but moved to the mainland to attend Willamette University for her undergraduate degree. She finds Piscine streptococcal pathogens fascinating and hopes by the end of the presentation you do too. And after the preview we got yesterday in the fish health section meeting, we're all excited to hear what you have to say. So take it away, Taylor.
Oh, good, because I'm excited to talk about it. So, like I said, I'm going to be sharing some of the work I've been doing, characterizing some of the agents of Piscine streptococcosis that have been popping up in the Americas. But I want to start by thanking all of my co-authors. As you can see, there are a good number of them, which I think speaks to the range and impact of this disease. Go forward. All right, so I'm going to start with a brief overview of Piscine strep and some of the bacterial pathogens responsible. Before going into the means by which I characterize them, multilocus sequence typing, and that'll bring us to the results of typing some of the more recently emerging agents of strep causing disease in the Americas. So streptococcosis in fish, it's actually caused by multiple species of bacteria. They're all what are called lactic acid bacteria, and so they share some similar characteristics. They're gram-positive, catalase negative, usually coccibacilli or bacilli. And the pathogenic ones cause sort of a similar disease presentation in fish, so they're all collectively grouped under the disease Piscine streptococcosis. Today I'm going to focus on just a couple of these agents. So Streptococcus iniae, which is probably the most widespread prevalent cause in fish, Streptococcus dysgalactiae, which is most often responsible for the disease in tilapia, and two more recently emerging bugs, Streptococcus dysgalactiae and Lactococcus garviae. So streptococcosis can manifest in a range of ways, but it's most commonly associated with outbreaks of high mortality, sometimes upwards of 75 or 80%. It's also a widespread disease. The agents of strep are considered to have a widespread global distribution, and their host range covers most hosted habitat types you can think of. Iniae alone is capable of infecting almost 100 different species of fish, and all of the ones we discuss also have zoonotic potential. In addition to causing these high levels of disease, they're also can be pretty tricky to manage. So as Dr. Yanong mentioned last week, it can be difficult to treat with antimicrobials, and we have the additional issue of we're seeing an uptick in some of those antimicrobial resistant isolates. There are no commercial vaccines for Piscine strep available in the United States. And while autogenous vaccines can be effective against a particular circulating strain, there's some really high interspecies diversity within the different species of strep. And that means vaccine escape is not uncommon. So altogether, this rounds up to this being a pretty expensive disease. It's estimated to cost the global aquaculture industry several billion. I think I might have even seen several billion US dollars annually. So the high interspecies diversity paired with the non-specific nature of the clinical signs of strep, and with the additional issue of some of those similarities that the lactic acid bacteria share, can make determining which specific strep is causing an outbreak difficult. So you can get a presumptive diagnosis made based off of the case history, maybe with some of these more typical signs of strep like exophthalmia, behavioral changes, spiraling, corkscrewing, lethargy, indicating the bacterial infection of the CNS. And then paired with isolation, pure isolation of a gram-positive cocci, that's catalase and oxidase negative, and possibly hemolytic on blood agar. particularly if you do it in anaerobic conditions. And then you can get a better initial ID to the genus or potentially species level using either biochemical tests like the API strep strips, although sometimes you might get a misidentification or non-identification, or single gene analysis like your classic 16S rRNA sequence, or there's been some qPCRs and LAMP assays developed for most of these species. However, these methods cannot get you to a solid conclusion on strain or genetic group, which can be really important when you're trying to make informed decisions for treatment or vaccination options. So to address some of those deficiencies, MLSA uses, or multilocus sequence analysis, uses multiple fragments of different housekeeping genes. So these are genes that are coding for proteins with conserved functions. So it avoids single gene bias a little bit more, but it gets you a lot higher phylogenetic resolution. It's also easily reproducible. You can upload it to databases, and vice versa. You can incorporate publicly available genomic information into your study. So it's a really nice system, and some people call MLSA and the closely related MLST the gold standard for typing bacteria. So we set out initially to develop an MLSA scheme for iniae. So iniae, we developed one with 9 different housekeeping genes listed there, and it resulted in a tree that gave us 5 different clades with 3 or more species, so A, B, C, D, and E, and those related back to some trends in phenotype, so the amount of capsule these different strains produced, and how hemolytic they were on sheep's blood agar. And so we published that scheme in, it was 2020, and in that, if you're interested in, we also saw that those clades further related to strain virulence in sort of a host-specific way. So the bacteria's ability to survive in fish blood from different hosts, tilapia, sturgeon, rainbow trout, and then also through a intragastric challenge of tilapia. We tested that MLSA scheme out on some other agents of Piscine strep that I mentioned, so S. agalactiae, dysgalactiae, and another one, S. ictalurae, which we will not discuss much more today, causes the strep in catfish. And it worked pretty well, which is exciting. So it was robust against multiple different types of streptococcal pathogens in fish. And I could also incorporate, human streps and things like that, so you can get a fuller picture of where these all relate to each other. So now when we get suspected strep, we incorporated this scheme into sort of our diagnostics where strep is suspected. So we have our suspected isolate, we sequenced those genes and made the phylogeny. And along this process, I ended up cutting TKT out of the scheme, particularly for S. agalactiae. It seems to work okay for iniae and dysgalactiae pretty consistently, but for some isolates of agalactiae, it seems like there might be multiple copies of the gene, which doesn't make it a very good candidate for MLSA if they're not conserved within their same isolate genome. Sorry if you can hear my dog barking. She's very angry for no particular reason. So this is what we get for a gigantic tree of all of our strep species. And this is too big to give useful information, so I'm going to break it down a little further. So we can see, if we look at our S. iniae and S. agalactiae strains, we have our 5 original S. iniae groups up here at the top. And we got some newer isolates of S. iniae from Brazil that interestingly formed their own sort of separate clade down here. So I'm still in early days and haven't done further investigation of what that means in terms of virulence, but it's definitely looks interesting. They're less distinct than the pygmy hippo isolates, which are so far the most unique strep iniae I've found yet, including in public reference data. So these guys could be a really cool next project to look at their phenotypes and virulence. And as for the S. agalactiae, we got some, just a few isolates of that. S79 also from Brazil in tilapia, and a strain from the USDA. And what's interesting about these, I'm so sorry, is that they group with isolates of S. agalactiae suspected to cause an outbreak of human cases associated with eating fish. So these were grouping with fish and human isolates from Singapore, rather than where I expected them to group with the other fish isolates that I'd previously sequenced or were previously available. So kind of interesting stuff to look forward in. S. dysgalactiae was almost more unexpected. So S. dysgalactiae is a less common cause of strep traditionally, but we've been working in collaboration with some folks at the USDA and in Uruguay. In Uruguay, it's been causing disease in farm sturgeon. So that's that group up top, A. And you can see that they grouped more closely with subspecies Equisilamus, which is traditionally associated with human disease. And we believe that this is the first sort of Equisilamus subspecies that has been isolated from fish. But even the USDA and also some isolates we're working with Dr. John Hawk on are grouping more distinctly from S. dysgalactiae subspecies than one would expect. So these seem to form really discrete clades that are separate from our established subspecies of S. dysgalactiae. And we started to do a little bit of work on looking at to what that might mean. And so Thu, an awesome postdoc in our lab, ran a challenge comparing one of those USDA strains and a sturgeon strain using a challenge model she developed for sturgeon with S. iniae. So challenge models for Streptococcus are a whole nother interesting tangent, but it can be tricky to find the right challenge model to mimic natural disease. And so she did an awesome study that found that intramuscular challenge following a period of stress is a great way to reproduce the disease. So that's what she did for the S. dysgalactiae, testing the different strains from different isolation sources in either sturgeon or tilapia. And so it looks like there's both a associated clade difference in virulence, and it's stronger in the host-specific manner. So our STC1 isolate from sturgeon was more virulent than our USDA strain, and it was particularly virulent in sturgeon, from which it was first isolated, causing almost, 50% mortality. So we got some nice lesions in those sturgeon, so that were consistent with what we've seen in sturgeon with strep before, so ulcerations in the muscle and skin, as well as internal lesions on the liver and spleen. And those are also reflected in our histopath. So Dr. Aline Henderson did our histo, and she made it awesome because I'm not a pathologist, so I'll do my best. But one of the more consistent changes was that we saw a decrease or absence of hepatocellular evacuation in fish that survived the challenge with STC1. And that's associated with a number of factors, including stress and disease, that reduction. Okay. We also saw at the injection site particularly necrosis and inflammation. There was disruption of the epithelium, particularly at that injection site. So when you looked further, you could also see the bacteria within those lesions. So both bacteria, STC1 and the USDA strain, caused some of these lesions where you could observe the bacteria present. But with the STC1 and more dramatically, it was also noted in other organs. So the kidney, heart, and liver, and vascular spaces also, you could observe those bacteria. So altogether, interesting start. Looks like there's some, again, a connection between our MLSA clades and genetic group virulence. So we're going to move forward with that. But because I like all of our agents of strep, we're going to talk about a cool problem, well, cool from a scientific standpoint, problematic from the fish's standpoint and those that manage them. A series of cases of lactococcus now. So our typical strep are all pretty closely related to each other. Lactococcus garviae is slightly more distantly related. It's still an LAB, it's still a coccus, but it more commonly causes septicemia that's more characterized with the presence of pretty intense hemorrhaging. So hyperacute hemorrhagic septicemia and particularly high mortality rates. And it's got a broadening host and geographic range. It's already been established as a pretty big problem for European trout farming. It's also an issue in wild marine fish farming in Asia, but it had previously only been sort of sporadically isolated in the Americas. There was an outbreak in the early 2000s in Brazil, but other than that, it had mostly been one-offs or, smaller scale. But more recently, it's been causing pretty devastating losses in farmed rainbow trout. So particularly there was around 2016 an outbreak in Mexico, and then relevant for us here in California at UC Davis, there was a big problem in some Californian facilities where we were getting, really heavy losses and isolating, this pathogen as the cause. And our isolates from these diverse hatcheries were particularly problematic. They were pretty resistant to the tested antimicrobials and treatment with fluorophenocal, which supposedly on the in vitro side might have been susceptible to, was still ineffective in the field. So difficult problem, this pathogen. It resulted in the culling of quite a lot of hatchery fish. So it's a big emerging problem. And in order to prevent that kind of thing, right, obviously we want to understand this pathogen better, understand its diversity and how that relates to treatment, control, and pathogenesis. So we tried to use our MLSA scheme to group these isolates and compare them to the ones from Mexico and existing isolates from other regions of the world. A majority of the genes selected for the streptococcal genus did not work for L. garviae, but some of them did. And an MLSA scheme generally is between 3 and 10 genes. And this one worked great with 4 genes. So we included the 16S gene, ARC-C, GRB, and RPOB. And we got a really nice tree that shows that our Californian isolates and our Mexican isolates seem to be really closely related. So by this, they were basically clonal, with the interesting addition of S69A. So if you see down at the bottom here, S69A is from what looks on a small screen, I'm sure, like a bowling ball, but is in fact a guinea fowl puffer. So that was from the East Coast. So it's not just West Coast isolates, which throws some interesting questions about transmission into the picture and how it's spreading. And it also groups with a published and tested isolate, PAC99, which is known to be particularly virulent. So we're calling this more of a hypervirulent clade. So this is a particularly virulent clade. And then we have some other groups of tamer virulence, we'll say, where we had some isolates we collected in surveillance for lactococcus, grouping with some of the known isolates from Asia and Europe. I also tested, single gene trees, because that can save on that initial first step where you want to start moving forward, because this is a fast, mortalities happen very fast with this disease, with Lactococcus garvey particularly. So the GRB gene alone was actually sufficient to get a pretty good tree that maintained those major groups if you just want to see where your isolate falls and if you need to anticipate it being one of those more virulent or less virulent strains to prepare to combat it. So the GRB tree might be useful for faster diagnostics, though of course you lose a little bit of resolution. We also did a pilot study looking for some differences in strain virulence associated to those clades as well. So this was run by Eric, who was a veterinary fellow in our lab and is now down doing his time in San Diego. So awesome job, Eric. We tested multiple different concentrations of an isolate from that Californian clade and an isolate from that was recovered from surveillance in California. And again, we see some pretty dramatic differences in strain virulence, with the isolate from that virulent clade clearly causing more mortality, right? They all but the lowest dose, which was only 10 to the 1 CFUs per fish, killed all of the fish very rapidly. So definitely moving forward, because this seems like it's going to be, so it's becoming, it's obviously becoming a problem. So our next steps are sort of focusing on these 2 emerging pathogens, with Strep dysgalactiae continuing to characterize those novel genetic groups. Some of our collaborators are doing some cool stuff with biochemically whole genomes that are adding to the oddness of these isolates from fish. They're not fitting in to the normal patterns of either established subspecies of S. dysgalactiae. I developed a live attenuated vaccine candidate for S. iniae, so we're going to look to see if it has any cross protection between species. And for L. garvey, similarly working on vaccine development for prevention, adding in new isolates and new challenges for biofilm disinfection and antimicrobial studies, and expanding on those comparative challenges in different host fish species. With that, I'd like to thank my collaborators and my laboratory and funding. Couldn't have done it without each and every one of you, and a special shout out to Teresa from the CDFW, who was a big part of isolating that and those initial L. garvey from California. Yeah, with that, I'll take any questions.
Awesome, thank you very much, Taylor. Great talk. We got time for 1 or 2 questions here, so either drop it in the chat box or feel free to unmute yourself.
Hi, this is Gary Marty. Taylor, just want to thank you for putting the pictures on the dendrograms for people like me who aren't used to looking at that. Really helps put things together. So just a compliment on your presentation.
Thank you. Yeah, I like drawing them when I don't want to do actual work, so that's good to hear they're useful.
All right, any questions out there?
Hey, Taylor, great presentation. I'll ask a question since it's pretty quiet out there right now. We heard also from Dr. Soto's talk yesterday how temperature is a big influence on this. Is that the case with all these Streptococcus pathogens? And how does that kind of correspond with the temperatures that trout are raised at in California?
Yeah, great question. Yeah, so with streptococcosis, sometimes people divide it into warm and cold water. So some of the agents are more likely to cause disease below 15 degrees Celsius, and the ones I talked about are more likely to cause it above 15. That doesn't mean they can't cause it, in temperate conditions or cold, but they are definitely more associated with having outbreaks in the summer months or when, you've increased the temperature of a facility in order to encourage faster growth. So with the sturgeon, for sure, and the trout, they're not always, so sometimes they're raised at slightly elevated temperatures than what might be preferable in order to facilitate faster growth. Or if you're thinking about wild species with the heat waves we've been having, definitely disease outbreaks are more of a concern there as well. So yeah, temperature definitely plays a big role, and it tends to increase the mortality rates with these agents for sure, and incidents.
Great, thanks. Sounds like vaccination is going to be really important to control some of these bugs.
Yes, yeah.
Hey, Jan, this is John Drennan from Colorado Parks and Wildlife. Hey, Taylor, great talk. When we've seen rarely we've got Lactococcus garvia in one of our isolation units in one of our cutthroat trout brood,
Okay.
but the water is only about 50 degrees. And so what we saw was like an inflammation of the gastrointestinal tract. And it was chronic low mortality if that's what it was, but you really didn't get to the disease except for that the intestines and everything else was really inflamed.
Yeah, so one of the things that I find really interesting about strep, and I mentioned just briefly that it can cause a lot of different disease presentations. So there is an association with chronic low-level streptococcal infections too, where you only lose maybe a couple fish every once in a while over a period of time. So, it causes potentially less economic trouble, but it's definitely still one of the ways that the different strains can infect. So I won't go too much into it for the sake of time, but in my strep iniae MLSA, like there has been an association with genetic group and whether it causes those chronic infections versus the more acute, infections as well. So that's interesting that it was the gastrointestinal system, because that's one of the proposed transmission routes is through the intestinal epithelium. So that's cool.
Yeah, that was that Lactococcus that we isolated from the Artemia zooplankton commercial feed.
Oh, okay, cool.
Yeah.
Yeah, so contaminated feed, very interesting. Good to know.
All right, well, thank you very much, Taylor. I hate to cut off the questions here. You got a couple more in the chat box if you want to respond to those once you're finished here, but virtual round of applause to you. Excellent presentation.